All right, good afternoon, everybody. My name is Aaron David. I'm the program director of plant ecology, and I'll be giving the plant ecology program overview and updates today, tag teaming it with the research assistants Sterling, Hannah, and Jeffrey. The purpose of this talk is really to give a broad overview of the work we do. So I'll be describing some of the program highlights from the past year and really emphasize how our data is being used by us, by our collaborators, and the broader scientific community. And then each of the RAs is going to go into in-depth on an ongoing project. So the 2024 plant ecology program team is, as I said, me, Sterling, Hannah, Jeffrey, as well as our interns, Sarah and Genevieve. Okay, so what do we do in the plant ecology program? Well, most of our work falls in one of three buckets. So we have conservation science, where our work informs understanding of how to best protect and conserve listed species. A bucket that I'll just generally call population ecology, where our work seeks to understand the fundamental processes and drivers of population dynamics. And then finally, biodiversity, where among other things, our work helps discover the unique flora and more recently, the unique fungi of the Lake Wales Ridge. And a lot of the projects that we work on really cut across these different categories, of course. And most of this work does take place in the scrub and the Lake Wales Ridge in general, although not every single project. And the core of what we do in plant ecology is plant demography. So this is essentially the study of population size, structure, and the underlying drivers. Many of our demographic monitoring projects date back to the 1980s, using many of the same tools that we use today, which are essentially flags, tags, toothpicks, and the occasional PVC. We are actively monitoring 15 species, including 11 species that are listed at either the state or federal level. And the long-term data that is generated from these studies is really invaluable, as we heard from Eric Mangus this morning, and as you'll hear in this talk. And I just wanted to kind of graphically or illustrate the work we do with these species. So they kind of come in tiers. So some of the species we work on are locally rare, uncommon, or what I'm calling super extreme range restriction. And then we have another kind of layer of listed species, including other species that are federally or state listed or Lake Wales Ridge endemics. And then beyond that, we have other plants that are endemic to Florida that we work on. So all but one of the species is endemic to Florida. And these layers of rarity really generate data sets that help us address different kinds of ecological or conservation-oriented questions. Okay, so the first part of the talk, I'll talk about some of the conservation science work that we do. And so we're talking about first evaluating introductions. So introductions are really important for the recovery of many species, but a big knowledge gap is how do we know if an introduction has succeeded? And then the second part of this section, we'll look at land management, specifically how we can improve land management practices to benefit rare species and their rare habitats. Okay, so like I mentioned, introductions are really conducted to help either establish or in some cases stabilize or augment populations, secure the persistence of the species. But it's a real challenge to actually evaluate how these introductions are doing. And because the plant ecology program has done so many of them in the past and continues to do them today, and monitor them after we do them, we're in a very unique position where we have all this long-term data that allows us to assess these introductions. So I'll just go through a few recent examples that have been published in the last year or are about to be published, hopefully. So this is a figure from a recent paper on Crotalaria avonensis, the Avon Park harebells. We did an introduction back in 2012, and we were able to compare 10 years of post-introduction data with a nearby wild site. And so in this case, I'm showing the population density, but we have similar figures and analyses looking at different vital rates. And so the introduced population is the big black dots, and then the gray bars are the distribution of the wild sites. And so you can see that beginning around like 2017, 2018, the vital rates were essentially equivalent to or exceeded the wild population. And this is one of the ways that we can tell that our introductions are actually succeeding. Another recent example comes from our work on Dicerandra christmanii, and this was led by Federico Lopez Borghese, who was a PhD student at UCF and is now a postdoc there. And so this study looked at wild population at the Flamingo Village Tract of the Lake Ridge National Wildlife Refuge, as well as an augmentation done in 2010 and an introduction to Carter Creek in 2012. And so what Fetty did was he developed a population model to simulate the time to extinction for these different populations. And so what he found is that although these introductions did seem to be more or less stable populations, they were predicted to go extinct 7 years earlier than the wild populations were. And we attribute that mostly to the underdeveloped seed bank that some of these have. So this can directly inform management of hopefully not burning them too frequently. One more example of this. This one is led by Hannah, who we're going to hear from later in this talk. So this looks at the success of Chrysopsis floridana introductions. Chrysopsis floridana is a federally endangered species that Bach Tower Gardens has been working on for a number of years. They conducted introductions in Southwest Florida, where it's found from 2008 to 2013. In 2017, we started getting involved with this project and looking at how they've been doing. And the real take home here is that even though all these introductions were done in introduced property or on protected properties, excuse me, only the ones that have really been given continued fire management seem to be thriving. So the ones that are burned recently, one of these, the laser, maybe the middle one. So the ones burned recently are increasing for the most part, and the ones that are not being burned are decreasing. And you can see that with the time since fire that the recent fires basically promote seed germination, and that is one of the reasons why it's so important to burn. This project is particularly important because in 2021, U.S. Fish and Wildlife proposed delisting the species on the basis of the success of these introductions. And what we're showing is these are not quite as successful as they look. And in particular, these populations probably need to be burned more frequently than previously thought. There it is. So we also can scale up some of our work to more global meta-analyses. So the plant ecology program contributes to the Center for Plant Conservation, or CBC's reintroduction database. Liz Gandy gave a nice introduction to CBC a few presentations ago. But this is just kind of a screenshot of the portal. But they have this database that basically tracks all these introductions, so we can go in and update and actually say how they're doing. And then they get used in meta-analyses like this one that came out this past year in conservation biology, which kind of aggregate across introductions worldwide. In this particular one, they found that the founder size and the habitat quality change are some of the best predictors of what introductions will succeed. For the land management side, of course, much of our work deals with fire, but we're also quite interested in how mechanical treatments can and cannot be used to also help manage our rare ecosystems. So one project which we are wrapping up is the population dynamics of endemic plants, or PDEP. The goal being to look at how different management tactics can influence the number of endemic plants. Some of the take-homes here are when you look at burning or hardwood reduction or sawing and burning, you generally see increases in endemic plants, particularly if you do the hardwood reductions first. But when you use these intensive measures like gyro tracking, which really mashes up the soil and does all kinds of really horrible things, you see a general decline in the number of endemic species, which is in part mitigated when you burn it. And I'll just make a note that while we're ending this project, we've been in full force with our new project across a lot of these FWC properties called the Community of Locally Important Plants. And the goal here is to really look at management and seeing how this affects the desired shrub community composition in these scrub habitats, particularly looking at how shrubs respond to burn seasonality and mechanical treatments. Okay, so here I'm going to hand it off to Sterling, who's going to talk about one project in particular, Elizabeth, and then Hannah will be following that with one of our land management projects.
All right, thanks, Aaron. Get this thing's on. All right, I'm excited to tell you about our Florida Ziphis project right now. So we just finished up our demography with this just a few weeks ago. So it's flowering right now. It's very thorny, so we get a lot of splinters during demography, but the flowers are quite nice and smell pretty good. So it has a lot of conservation considerations. It's one of the rarest of the rare. So it is a Lake Wales Ridge endemic, and it's federally endangered, state endangered, and a G1 on the NatureServe rating. And we know of 13 extant wild populations currently. A few of them have gone extinct over the years. So you can see the range map here on the right. The red ones are the wild populations. And so while that's a decent number of populations, most of them are highly clonal, and most of them also have fewer than 30 plants, which are just clonal ramets. So because of that, we have done several introduction sites across the ridge, which are in blue here. And those are helping to kind of improve its outlook. But it does have reproductive limitations, so it's an obligate outcrosser, and only 3 compatible mating types are known that are able to produce fruit. So just a brief shout out to all the people over the years that have really laid the foundation for this project, especially Carl and Eric and many of the RAs and interns that have come through. So I'll just talk about a few of the recent developments in the project. So we had our Florida's Fish Recovery team meeting right here, actually, at Archbold just this last May. So we were able to engage with 17 people from many different organizations. So we were able to do a lot of strategic planning with a lot of different stakeholders and organizations from across the ridge and also across the country in some cases. So this is integrating different kinds of ecology, genetics, conservation, and horticulture. And so I'll talk about some of those collaborations. So one of the biggest, most important ones for this project is the genotype database. So we've been working on that with Dr. Christy Edwards at the Missouri Botanical Gardens. And so she has helped us identify 50 total genotypes, 46 are extant right now. So this figure just shows the distribution of the Ziziphus genotypes across all populations. Gray are the wild, blue are introduced, and pink is just the captive ones at Bach. So as you can see, most of the wild populations are really genetically depauperate, except for a few of them. But with our introductions, we do try to introduce more genetic diversity. So knowing these genotypes is really key to determining which ones are cross-compatible and able to produce offspring. And some more interesting work by Dr. Bridget Williams in St. Louis showed that epigenetics, so these are factors outside of the DNA, may also play a role in the mating type. So this figure is essentially showing the different mating types of Florida Ziziphus in PC space and showing that they're differentiating epigenetically. And this may ultimately lead us to potentially some genetic markers that we've been in search of that could be controlling self-incompatibility. So another important project that we've collaboration is with propagation. So we've worked with Dr. Valerie Pence at the Cincinnati Zoo and Prue, that's been mentioned. So she's been helping us with the asexual propagation of Florida Ziziphus stems. So this is an additional way we can create plants for introductions. And so she's able to culture these stems in this media and create entirely new Ziziphus plants from them. And Cheryl Peterson at Bach Tower has also been doing germination trials for many years. So we've learned a lot about the seed ecology of Ziziphus that way. And importantly, this provides a lot of propagules for our introductions. So speaking of introductions, we just put a Florida Ziziphus introduction here at Red Hill just this last August. So we were able to plant 73 of these germinants there, including 13 unique genotypes. And thus far, we've had 100% survival, which is pretty good for Ziziphus, yeah. Knock on wood. We've also had a seed introduction project, which we started in 2021. These are kind of excess seeds from Bach Tower, but we've been able to sow about 14,000 of the seeds. And so we've been able to do that at Carter Creek, Flamingo Villas, and now up at Red Hill. And so it seems like, at least for now, the Ziziphus is preferring to germinate in closed canopy sandhill habitats. And we actually just got 5 germinants up at Red Hill just recently. So just to summarize this work, our ultimate goals with this project are to protect the existing wild populations, but fundamentally these wild genotypes are kind of the currency we're thinking of. And these can be used to promote and to restore the self-sustaining and ecologically functional populations, whether those are in the wild or introductions. So this is really a multi-institutional collaboration, and it's one of the rarest of the rare, so we'd really like to keep it hanging on and hope to do that by using these cross-compatible genotypes. So with that, I will hand it over to Hannah. Have a mic.
Yes.
There you go.
Thank you, Sterling. Hello, hello. I'll be sharing about rescrub, which, as you can see from the title of this slide, it's a nickname that we have for a U.S. Fish and Wildlife coastal habitat rehabilitation project. And the idea is that we are restoring disturbed rosemary scrub, which actually Kevin gave a little introduction to, as well as Jenny Schaefer in her talk about rosemary balds or rosemary scrubs. So over on the left is a picture of what a disturbed rosemary scrub might look like. They're more open. We've got it mostly dominated by Ceratiola ericoides shrubs. They're not rosemaries at all. Actually, they're in the ericaceous family. And either these shrubs are too far apart so that they can't actually really carry a strong fire, or there's too many oaks in between, which we know are fire resistant. And so then what ends up happening is this ground cover of Selaginella, kind of like a club moss, like a podium moss, and lichen, which sort of just takes over on top of the surface of the sand and makes it harder for endemic germination to occur. So we'll see in a disturbed rosemary bulb a lack of herbaceous endemics. And then here, contrasting on the right, is a picture of a more functional rosemary bulb ecosystem. So we've got rosemary shrubs that are close enough for us to carry, and then, as we know, there's a lot of herbaceous endemics present in this habitat type. And so with rescrub, the question that we are investigating is mowing before a burn beneficial for a degraded scrub habitat? And we're using as the metric the response variable in this germination of endemic seeds. And so here we have on the right some Eryngium cuneiformium that has since germinated. I'll tell you more. And so, as Kevin mentioned, he so skillfully mowed within a section of scrub out in the reserve. He was able to mow in 40 by 40 blocks, only areas that we had designated. And so he ended up mowing about 66 40 by 40 blocks. And then we, out of those, selected them for which were going to be our experimental plots. The greens were blocks that were mowed, and then the brown ones were unmowed. And then in this experiment, we took advantage to test a couple of other factors that might affect germination. So in this experiment, we are testing for mowing. We're testing for burn because the way that this fire happened, not all of the blocks were burned. We're testing for fuel class. So in the blocks that were mowed, we created these mow piles, little mow debris piles on top of some of the plots. And then in all of them, both the mowed and the unmowed, we're testing for whether the seeding plot is next to a shrub, whether it is in bare sand, like this person here, or whether it is in on top of either naturally occurring ground cover, like the Selaginella and Gophrabula I spoke of, or a handmade debris pile. So within that also, we're testing for shrub type, with the shrubs being oak, rosemary, and palmetto. And so each seeding plot, we had 216 total. Each seeding plot got 40 Eryngium cuneiformium seeds, 10 Hypericum cunelicula seeds, and just 2 Beatrice olingerae seeds. Actually, folks from Fish and Wildlife came out and helped us with the seeding, which was really great. And then here you can see some drone photos of the experimental site before and after the burn. And as I said, we were testing for a couple different things. So we were testing for mow treatment, whether it was burned or not, the fuel class, and the shrub. And so with the germination of each species that we seeded and other endemic species of interest that we did not see, we're having that be the response variable. So on the left, we have just a bar graph of total germinants on the y-axis. And then the colors indicate the fuel type. And so both in mowed and unmowed, you can see that the bare sand plots had the most germinants. However, wasn't statistically different between mowed and unmowed. And so that's really the only difference that we have found so far. Here on the right, these are estimated marginal means graphs. And then when the red is not overlapping, that's when it's a significant difference. So as you can see, the bare sand plots on these were overall higher in a significant way. This is for Eryngium and Liatris. We haven't seen any Hypericum germination so far. But so that's to say is maybe the reason for this is because all those bare sand plots perhaps received less fire, so weren't scorched as heavily, and we're gonna see germination, but it's just taking some time. I'm sure some of the legendary plant people with us could also hypothesize about that. But this is an ongoing experiment, and oh yeah, it's this other result that we happened to take data for was we were looking for the presence of invasive Natal grass in both mowed and unmowed plots, and we found no statistical difference between those two. Also, the effective burn was not significant, so that's really good news to know that at least this mowing treatment is not harming this ecosystem and not creating another problem. So in summary, with this ongoing land management study, we are revealing which practices most favor endemic plant germination and rosemary scrub restoration. And I will hand it back over to Erin.
All right, thanks Hannah and thanks Sterling. All right, so that was the conservation science portion. So we'll talk a little bit about the population ecology. So work in this area really informs our fundamental understanding of population dynamics, as well as particularly with the long-term data sets informing our understanding of how species are responding to climate change. And so I want to highlight a few papers that have come out or that we're working on. The first is one led by Warren Abrahamson on what kills the virtually immortal palmettos. So the palmetto is a real foundational species here in the scrub, and it's also incredibly long-lived. Many of these individuals can live hundreds and in some cases thousands of years. And so Warren Abrahamson started the Palmetto Project in the early 1980s, I think it was 1981, and 1980, anyway. So he has a long-term data set, and we're really interested in looking at how we could use this data set to understand how frequently these things actually die, and then what might be killing them. So some study highlights. Over 42 years, these have exceptionally low mortality. 5.7% of the saw palmettos and 3.5% of the scrub palmettos died, so not very many. Culprits the first one was extreme fire. So these were very hot fires that often were escaped or prescribed burns, but that burned very hot and actually managed to kill palmettos. And in some cases, the opposite, a lack of fire shaded out palmettos and led to mortality. What doesn't kill palmettos, fortunately, we didn't see any role of disease, particularly some of the emergent palm diseases that we're seeing in this state. And we didn't see any effect of drought on palmetto death. So the take home from this study is that even these virtually immortal palmettos need referral management. So I want to mention a few other interesting studies that have come out this year, advancing ecological theory. So this one by Hindle et al. Used our Eryngium data set and had this very unique modeling approach to looking at climate drivers. And they found that Eryngium is actually one of the species that might benefit from climate change. And it seems to be adapted to the future predictions of what climate is going to look like. And importantly, it might persist under a range of fire regimes. So it's not going to be quite as tied to the fire as it has traditionally been. There it is. Another really interesting study that came out this year that I just wanted to mention to you all. So this study that was in Nature looked at a new theory for why species coexist with each other. And the theory is basically that species with complementary life history strategies are able to coexist and not compete one another. And in support of their theory, they took some of our long-term data from 6 scrub species and found that they are more complementary to one another than would be expected. And that basically supports this new theory of why species coexist. And then lastly, I'll mention a new study that's just starting on the role of microbes in regulating plant populations. So this work is being led by Josh Fowler, who is a postdoc at University of Miami, working with Michelle Afkami and Chris Searcy. Josh is kind of following up on work that I did when I was a postdoc at University of Miami, except he is looking at a much bigger experiment. He's looking at the role of soil microbes found in 30 rosemary bulbs across 11 different species. So he's quantifying microbial effects on germination and growth, and then incorporating those into population polls using the Archbold long-term data. And so one of our new areas that we're starting to get into is trying to integrate our plant demography with plant traits. And Jeffrey is going to be talking about a new study that we ran last year. I hand it over to you.
You. So yeah, plant traits. Traits influence a plant's ability to survive, and their survival on an individual level influences the population and its demographics in a larger scale. So we want to see if we can find traits that vary within individuals or between individuals, and traits that vary between habitats. Are they using different strategies to survive in different habitats? And so a lot of research has been done in plant traits recently, and researchers have kind of identified a couple of axes that a lot of traits correlate on. One of them is a slower growth strategy versus a faster growth strategy. How much they're investing in their leaves to make something that's like going to live a long time versus just growing as quickly as they can to get out as much surface area. And so the variable we'll be looking at for that is a specific leaf area, which is basically how spread out it is for its weight. And then we have a collaboration versus do-it-yourself axis, which has to do with how much they're relying on mycorrhizae to scavenge nutrients for them versus how much they are growing their own fine roots to do it themselves. And so the variable we looked at for this is specific root length, which a high specific root length would mean fine roots, which means they're mostly actually scavenging their own resources rather than relying on mycorrhizae. And so we compared several species in scrubs and in our sandy fire lanes. We hypothesized that in the disturbed fire lanes, we would see a faster growth strategy and a more do-it-yourself strategy, since the soil would be disturbed and maybe there wouldn't be as much mycorrhizae there. And faster because it's a disturbed habitat, and so they kind of maybe have to capitalize on what space is available. So these are our 5 species. They're all in rare scrub species. This here is a root of euphorbia. We looked at their roots, we looked at their leaves, we got these data, specific leaf area, specific root length, as well as a few other things that I'm not going to talk about too much today. We did this all in the summer. I believe most of you are already familiar with summer field work, but here's a little graphic to help you understand if this isn't something you've experienced. We analyzed the image. We analyzed the roots and leaves using a couple of software. Hypericum had really small leaves, but we managed to get our SLA for it. So speaking of this fast versus slow gradient that I spoke of earlier, we saw that habitat is a significant predictor. Also that species habitat interaction is significant. So different species are responding to different habitats in different ways. So we see some are just about the same in scrub versus road. Dicerandra in particular has a lower SLA in the roads, which means we're actually seeing a slower growth strategy in the roads, which is the opposite of what we had anticipated. And there could be a number of reasons for this, but it's all hypothetical for now. In the collaboration versus do-it-yourself gradient, habitat was once again significant. We don't see any individual species being significant here. That is because this is just a pilot study and we don't really have that many data points. But we do see general trends towards higher specific root length in the roads, which would suggest fine roots, they're doing their own work. It's a do-it-yourself strategy. And We found the highest coefficient of variance in specific root length, so that may be a variable that's good to look at in the future because it varies a lot between individuals. So we're going to skip the recap because I'm out of time.
No, I think we're all out of time actually, but real quick, for biodiversity, just a single slide, I want to mention that we are discovering new fungal species, working in a new collaboration with some folks at Rutgers, and so we're gonna have some new species descriptions for you. That's all on biodiversity. I'll wrap up. No, I won't wrap up. Basically what we said: conservation science, population ecology, biodiversity. That is what we're doing. Acknowledgements, lots of people, especially Eric Mangus, who set up many of these experiments and long-term studies. And I think we're out of time for questions.
Oh, we could take a question while. Thank you. Yeah. Maybe while Laura's getting ready to come ahead. Great.
The question was on the re-scrub of, we looked at ones that were species that were there versus ones that we sowed. The ones that we sowed generally were not there. So Orangium wasn't there, Oleatrus wasn't there, but we were also taking note of which ones we were finding. So the only one that we sowed that was there already was the Hypericum, but we didn't observe any of that, at least not yet. So there really was no Orangium in that site.
Thank you. And I think we're ready for our next talk. And the moral of the last one is don't be the last leg on a relay.
All right, we have you on our screen here, so you can start.
All right, thank you very much. Can you see my screen okay? Okay, I hope so. Let me know if you can't hear me or see my screen okay.
No, we're good in the meeting room.
Oh, awesome. Sorry, I just can't see any of you. But hi, my name is Shailee. I'm a postdoc currently a postdoc at Cornell, but this is work I did as a postdoc in Nancy Chen's lab at the University of Rochester. It's still ongoing, so I would love your feedback. But yeah, I'll be talking a little bit about my project linking limited dispersal, inbreeding, and lifetime fitness in Florida scrub jays. And just thank you in advance to Sarah and Christopher for setting the stage for Florida Scrub Jay talks. I'll be going over some of that basic information again, but thanks for that. So just to start out, what is inbreeding? And we actually talked a little bit about this today too. So inbreeding is defined as situations in which matings occur among relatives and the associated increase in homozygosity, which is when individuals have the same two alleles for a gene that results from such matings. So here on this pedigree, for example, you can see I've circled the matings that are resulting in inbreeding in the red circle. And inbreeding is known to have negative fitness consequences, which have been documented in even in wild populations. So I'll just go over slide advancing. It's a challenge. I'll just go over some classic examples of some of these studies. So for example, inbred stowaway sheep from an island population have been shown to be more susceptible to parasite infections. Inbred song sparrows, also from an island population, have been documented to be more susceptible to mortality in storm events. And inbreeding in red-cockaded woodpeckers has been shown to result in lower hatching rates and lower survival rates in the offspring. And so because inbreeding can have such drastic impacts on fitness and even in wild populations, it's important to understand the causes of inbreeding. And so the two main sort of ways in which inbreeding can arise in a wild population are, first of all, if it's just a small isolated population where even if the individuals are undergoing random mating, there just aren't a lot of opportunities to mate with non-relatives, and so they just end up mating with close relatives. But second, there could be a sort of behavioral component in which, in addition to that sort of small isolated population, you might have some sort of non-random mating in which individuals with similar genotypes are more likely to mate than expected by chance. And so it is this second scenario, which can be further broken down into either a direct preference for individuals with similar phenotypes. This is called phenotype matching. And this has been documented in, for example, some species of cichlid fish, where they prefer to mate with individuals that look similar to them, and that results in inbreeding. But then there can also be some completely different behaviors that seek to maximize short-term fitness, but in turn they lead to inbreeding sort of as an indirect effect. And this inbreeding can be tolerated on some level in the population because of that increase in fitness that presumably outweighs at least the short-term costs of inbreeding. And so it is this second behavioral component that I'm interested in exploring today. And so a quick introduction to the study species. So though they have been introduced already, today is Florida scriptrays. So Florida scriptrays are a cooperatively breeding bird that is endemic to the scrub habitat in Florida. They hold territories year-round in the scrub, and so that makes them quite amenable to long-term studies. And there has been a very long-term study of Florida scriptrays at Archbold since 1969. And just as a note for the Avon ecology folks in the room, today I'll be talking about data from 1990 to 2021, and I am focusing on individuals that were born within and or dispersed within the demo tract. So the study is possible because of the extensive fieldwork undertaken by the Avon Ecology Lab at Archbold, so thank you. I am simply playing around with the extensive data that they've generated over the years, and this data is really impressive. So because each individual is uniquely tagged with color bands and followed throughout its life, we have a very extensive complete pedigree, which we can use to calculate relatedness between individuals and also track their reproductive success over time, over their lifetimes even. And then there's also, we have very accurate data on dispersal distance, territory maps, and monthly censuses. So we know who's alive in the population every month. And so, of course, over the years, this data has led to many insights in the Florida scrubjay population genetics, demography, and behavior. And I'm building off of some of what we know already today. So we know that in Florida scrubjays, both sexes disperse and females disperse farther than males. And this generally leads to, but both sexes are sort of limited in dispersal. So this leads to a buildup of what we call a kin neighborhood, where related individuals are found closer together in space, especially for males. Both sexes generally disperse to establish as breeders and stay breeders until death, though they can sometimes switch mates. And the Archbold population specifically is becoming increasingly isolated because immigration has been documented to be decreasing over time. And also using genetic data, Nancy Chen showed in her 2016 paper that there's evidence of fitness reduction due to inbreeding depression in this population. And we also know from careful behavioral ecology studies, such as Fitz and Reed's in 1999, that there's strong preference for short distance dispersal in both sexes, presumably as a result of higher fitness due to familiarity with the environment when individuals don't disperse too far from their natal territories. And so for this project, I'm interested in asking the following questions. So the first question is inbreeding in the Archbold population sort of exacerbated by behavior, specifically by short distance dispersal, because that might lead to non-random mating with closely related individuals? And then second, is there any evidence of inbreeding avoidance? So if you're not dispersing too far away, do they in addition have some sort of strategy to avoid breeding with closely related individuals? And then lastly, do the fitness benefits of short distance dispersal outweigh the fitness costs of inbreeding in this population? So I'm going to quickly walk you through my random mating model, which is the main method that I used to generate results for this project. What I did was I took the observed data, which looks something like this. And for each year, we have all these different territories. So that's the fourth column on this data set. I don't know if I have a pointer, but the fourth column. And you can see that the territory is the year of the territory as well as the name of the territory, so that I've just reduced the names to 1, 2, 3 here for simplicity. But you can see for each territory we have a pair, so the male is X and the female is Y. And for both sexes, we know the previous territories that these birds came from. And also, in this sort of cartoon example, you can see that male X actually inherited his territory. So he was born on territory 1 in 1999 and then became a breeder there, presumably sometime around 2002. Okay, so what I do in my random mating model is I take this data and first I shuffle all the males around. So here the two colors represent the columns that sort of go together in this simulation. And so you can see that the males have been switched around. And so their associated previous territories have been switched around. But the females have remained stationary and remain associated with the territory that they were associated with in this simulation. And then I similarly do the same thing for females and keep the males like stable and stationary and associated with their territory. And then I do this like 1,000 times to generate a random a simulation of random mating in this population for every year that we have data for. And then not only do I calculate relatedness of these simulated breeding pairs, but I also calculate dispersal distance. So we have territory maps for all of these birds. And once I do these sort of random shufflings, I then simulate dispersal trajectories for all of those random matings and calculate dispersal distance, which for Florida scrubbees, we calculate dispersal distance as number of territories crossed for dispersal. So this is very quick, and if people have more questions, I can talk about it more after the talk. But now I'm going to just dive into some results. So the results will look something like this. So here on the x-axis, we have year. And on this particular graph on the y-axis, we have dispersal distance, which, as I mentioned, we measure as number of territories crossed during dispersal. And that actually correlates very closely with actual dispersal distance in meters. And then here, the gray cloud of data points are results of the random mating model, which there's a lot of them because I run the model 1,000 times. And then the black points, sorry, that's my dog. And the black points are the observed mean and standard error from the observed data set. So if it's a filled circle, the value is significant. And if it's an empty circle, the value is not significantly different from the random mating model. Okay, so here I was testing for shorter dispersal distances than expected by chance, and that's exactly what I found. So you can see that for both sexes, there's a strong preference to disperse shorter distances than you would expect in a random mating model. So that's interesting, and that kind of that is what you would expect based on previous studies in this species. And then I looked at relatedness of these observed breeding pairs and compared them against relatedness of simulated breeding pairs in a random mating scenario. And so here I'm testing to see if the relatedness that we observe is ever significantly lower than expected under a random mating model. And this is a lot of plots, but the main takeaway is none of the circles are filled in. So we never observe lower than expected relatedness between breeding pairs in the Archbold population. But instead, if I take that same data and then I test to see if the relatedness of breeding pairs is ever higher than expected under a random mating model, I do find several years in which relatedness is higher than you would expect. So the first sort of row of plots is mean relatedness of the breeding pairs, and the second row of plots is the proportion of close relative breeders amongst all the breeders in a given year. So it's the same way, or it's like two slightly different ways of looking at the same data. And you can see that the data aligns for most of the years in these two slightly different ways of looking at it. So it suggests that this higher than expected relatedness that we're finding for some of these years could be partly driven by pairings between very close relatives on the order of parent offspring or siblings or half siblings or even first cousins. So sort of driven by the short distance dispersal that I found, that I showed you earlier. So to sort of summarize that a little bit, there seems to be no evidence of inbreeding avoidance, and there even seems to be some evidence of tolerating inbreeding, which arises due to short dispersal distances. And so the question then is, how do fitness costs and benefits shake out, right? Do the benefits of short distance dispersal outweigh the costs of inbreeding? So I'm going to now show you some results from the long-term data that will look something like this. So on the y-axis, there is fitness. Here, it's lifetime reproductive success, which we can measure using 3 different metrics. So survival of an individual's offspring to the fledgling, independent, and yearling stage. And then on the x-axis, it will change for different plots. But for this first result, I'm showing weighted relatedness to all social mates. And so this is the relatedness that an individual has to its social mate, weighted by the number of years that it was in that breeding pair. And this is summarized for all mates over its entire lifetime. And so the first result I'm showing you is what we would expect from previous evidence of inbreeding depression in this population, which is that the lifetime fitness of both sexes is lower if they're in breeding pairs with closely related social mates for long periods of time. However, what is interesting is that if you only look at individuals having a closely related social mate at some point in their lifetime, so if you take that time component out, it doesn't actually impact their lifetime reproductive success. So here, blue is individuals that have been in a pair with a close relative at some point in their lives, and black is individuals that haven't. And you can see that there's no significant difference in their lifetime reproductive success. So it's really that time component, but if you end up in a breeding pair with a closely related mate, that doesn't automatically tank your lifetime reproductive success.
Heads up, 2 minutes.
Oh, wow. Okay. Okay, going back, sorry. So going back to this graphic, there do seem to be some costs of inbreeding. And then I'll just blaze through some of the benefits of short distance dispersal. But what I found was that both sexes do get to breed for more years when they remain close to their natal territory. They don't vary in their likelihood of survival, but I'm going to skip this for right now because it's a little bit complicated. And the main result is what people have found before, which is that lifetime reproductive success for both sexes is higher at shorter dispersal distances. So going back to this plot, there do seem to be some benefits that are outweighing the costs of inbreeding. And in the interest of time, I will simply summarize the rest of the findings, which is that we found that both sexes do disperse shorter distances than expected under a random mating model. Crucially, we found no evidence of inbreeding avoidance, which is interesting. And we found that having a closely related mate actually doesn't significantly decrease the lifetime fitness of an individual if you take that sort of breeding pair duration component out of it. So in and of itself, it is not a strategy with low fitness if you end up with a closely related mate. And as studies have found before, short distance dispersal leads to higher lifetime fitness for both sexes. And surprisingly, this is a result I skipped over, but we found that individuals are not significantly more likely to switch mates when paired with a closely related mate. So there doesn't seem to be some sort of coping strategy for ending up in a closely related breeding pair. So overall, these results show that short-term fitness benefits can lead to long-term fitness costs, as evidenced by this population of Florida scriptures. And the big takeaway, I think, is that individual level fitness optimizing behavior can sometimes negatively impact population level processes. And with that, hopefully I didn't go too over time. I will just thank everyone and happy to take questions over email if I don't have time for questions right now. Thank you.
Thank you. You caught up perfectly. Thanks.
Yeah, thank you. My timer was not working, so that was a useful heads up.
Yeah. We could. We're moving on to the next speaker. Are you ready to go? We'll take one. If you're still there, we could take one quick, like 30-second question if anyone had one.
Yeah, sure. Let me check is there, yeah, no questions on Zoom that I can see.
I don't see any, so we'll move on to Tiffany now, but thank you so much for joining us and very
No
good
problem.
talk. Thank you.
Thank you.
Okay, so I'm Tiffany Doan, and I'm first range. Okay, they can still hear me, right? Okay, so, well, not going anywhere yet. So I have a nice title, but I really am not going to be able to answer the question that I put in my title because we're pretty new in this project. So I'm mostly just going to give an overview of what we've done so far, and then just the small amount of results we have, and then talk about what comes next. There we go. Okay. So this is the SERDIP project, which is Strategic Environmental Research and Development Program. And it's a 4-year project that several of us are doing. You can see the collaborators here, some from Archbold, from UCF, and from Reed-Noss from Conservation Science. And so the DoD gives these grants for particular solicitations to conserve threatened and endangered wildlife on their installations. And our solicitation was specifically for multiple stressors and how they synergistically combine to affect species. So you're probably familiar with Avon Park Air Force Range, but no one's done a talk on it yet today. So it's in Polk and Highlands County, a massive installation, 106,000 acres, about, I don't know, half hour or so north of here. And so the project is trying to look at these multiple stressors that I'll talk about in a minute. We're doing all of the upland habitats found at Avon Park. So pine and flatwoods, dry prairie, Florida scrub, and then the ecotones between those habitats. So the multiple stressors that we selected to work on are invasive species. So fire ants, everyone is familiar with those. Fire ants, we know, are bad for ground nesting wildlife in particular because they kill any baby animal that hatches out of an egg on the ground. They can go kill it. And so we know this is well documented, but what we don't know is how that is it works sort of synergistically with these other factors. So another stressor is a natural one, and that's burning of the habitats. All of these habitats burn naturally at varying frequencies. And the Air Force Range is very good about burning, unlike many other places. They're very active in burning, maybe too much in some cases. But that is a stressor that is good, of course, to some species, but how does that interact with fire ants is one of the questions we want to look at. And then our third factor is grazing. And the grazing project is only going to be done at DeLuca. So I am not the herpetofauna, the part that I'm doing is not involved in that, so I won't speak about that anymore, but the ornithologists on the project will be doing some work at DeLuca with grazing as well. So it is a 4-year project. In the first year, it's really just a survey. We're seeing what's there, what's where, as far as threatened and endangered wildlife and also fire ants. And starting next year, and next year is this fall, we'll start our 2nd year, we'll be doing the experiment where we're trying to look at all these factors, so habitat, fire ants, and fire history. And we're trying, as I said, to look at the synergistic effects. So how do those all combine to affect the wildlife? So the species that I'm working on are the ones listed here. So we have eastern indigo snakes, sand skinks, and blue-tailed mole skinks. Those are the federally threatened species. And then the state threatened species that we're also working on are Florida pine snakes, short-tailed snakes, gopher tortoises, and gopher frogs. So that's the parts that I'm responsible for, but we're also studying 2 species of threatened mammals, and then several species of threatened or endangered birds. So and sort of indicators for each of the habitats, like Florida grasshopper sparrow in the prairie, Florida scrub jays in the scrub, and red-cockaded woodpeckers in the pine flatwoods, and then several other species. And as I said, I'm not going to talk about those today, but they are part of our project as well. So for the herpetofauna part of the project, I have 60 plots spread around the range in all of the different habitats, so 12 per habitat or ecotone. They're 1 hectare plots. And each plot in the very center has a trap that we installed, which is a drift fence array. And I'll show you pictures in a moment, but it's a triad array with 3 10-meter fences. And the fences are countersunk 15 centimeters, so that the idea is that an animal will be going along either above ground or underground, run into the fence, and the natural behavior of most animals is then turn and then follow the fence to the end. And so that works for both species that are fossorial and also those that are on the surface. And the traps are divided into 2 different types, half and half. Some of them have pitfalls, so buckets, and the other ones have cameras. So I tried to make a map that you could see, but I can't because it's such a big spatial extent that you can't actually see anything. But there's the range, and you can't see it at all. But you can kind of see that there are little boxes here. You can see some of them. Anyway, it's a very big spatial extent, so there's no way to put it all in one map that you can see anything. But we have traps along the South Ridge, in the prairie flatwoods ecotone, prairie areas, the 3 main prairies, and then up in a lot of flatwoods and some scrub and scrubby flatwoods up there. So a large part of the range we have traps. Putting in these traps was a ton of work, and lots of students were tortured in the sun by doing lots of digging. We weren't allowed to use any mechanical equipment because it's an active bombing range, so we couldn't run into bombs. So we used shovels and axes to install these things. I couldn't have done it without lots of students. So the two types of traps that I have are, all of them are the drift fences, but one type, half of them have they're pitfall traps. They have buckets in the center and at the ends of the 3 arms. And additionally, they have funnel traps, so that's a total of 4 buckets, 19-liter buckets, and 6 funnel traps. I sample for 8 days, twice each quarter, so every 6 weeks. 8 days the traps are open. Traps have to be closed when we're not sampling so that something doesn't fall in there and then die before I can get to them. We have to check them every day. And so this is what they look like. So you can see the triad. Here are some funnel traps along the side, and then you can't see the buckets, but there's a bucket there, and there's a bucket at the ends of each of the traps. And so the animal will fall into the bucket or slither or walk into a funnel trap and wait there until I take it out the next day. The other ones are camera arrays. So instead of having the buckets, there is instead an upside down bucket with a camera attached that's shining down. It is custom focused to 28 centimeters so that it can take a picture of whatever animal goes into the bucket. And this is not a great picture, but this is upside down looking down into the bucket. So the camera's attached. There are cutouts, 3 cutouts, 1 for each fence. And so the animal will go along and then go in and be directed under the camera and have its picture taken. And I call it a trap, but it can just leave. It's not actually trapped. And so that's what those look like. So you can see the 3 fences, and then there's that upside down bucket. Unfortunately, we only have 1 bucket per trap because the cameras are very expensive. So only the center has the camera. The ends do not. The third method is gopher tortoise surveys and cameras. So each of the 1 hectare plots, I've done 3 100-meter transects to look for burrows. And we use line transect distance sampling. And for this project, we're most interested in the juveniles because it's a 4-year project. Gopher tortoises live a long time. So we're probably, no matter what happens with our experiment, we're probably not going to see changes in the adult gopher tortoise populations. I guess we could, but probably won't. But we could see changes in the juvenile populations because they are directly killed by fire ants, and also they're growing and changing and moving. So we'll be able to see those. Additionally to the burrow surveys, I have cameras at the mouths of burrows that you can see anything that goes in and out of the burrows. And so not a great picture, but here is one of the cameras. There's a stake with the camera. The burrow is over here, can't really see it. And then these are just some interesting burrows. So this one, active burrow, you can see the plastron marks and going in there. And this is one of the most interesting ones. Again, hard to see. It's actually an adult burrow, but there is a little trail of a tiny little juvenile. It's very clear. You could see its feet, and actually I could measure the size of the tortoise. So hard to analyze it because it's using an adult burrow, and it's a tiny little tortoise, didn't dig its own burrow. We can see any of the tortoises. We can also see all the commensals that are going in and out. So far, we haven't gotten that far. I've had 3 pitfall sampling trips, so that's 24 days. I'll start another one next week. And 52 days of camera footage so far. So again, we're very early in this. And so far, we've gotten 26 herpetofaunal species, which I'm pretty happy with for the effort we've had so far. 22 of those species we've gotten pitfall traps, and 12 in the camera traps. And then plus 5 bird and mammal species that I've gotten in the camera traps as well. Camera and the buckets, so camera traps and the buckets. But the whole purpose of the study is for threatened and endangered species, and so far I've only had 2 in threatened individuals, 1 indigo snake and 1 blue-tailed mole skink. So those are really hard to find, kind of a needle in a haystack thing. So hopefully we'll be able to see more of those as we continue. As far as habitats, we've gotten animals in all the habitats. These numbers are strangely inflated because of certain individuals. So we actually have the most probably individuals in the scrubby flatwoods, but we have some mice that are probably the same individual that go and come into the cameras every 30 minutes of the same mouse over and over and over again. So because it's not staying there, I have to consider it another record. And so that's why we have in the scrub, 1 million mice, but it's probably just a few individual mice that are doing that. And then in the prairie, we have skunks that spotted skunks that do the same thing, go hang out on the cameras, literally hang out, lay down, get its picture taken multiple times. So here's a list of species we've found so far. The red ones are the threatened species, but we've had a pretty good diversity. You can see a few pictures. So oak toads are our most common ones that we found. That's a pygmy rattlesnake. And then here is a picture from the camera so you can see what it looks like. That's a scrub lizard, which are fairly common. But the camera is facing down at the lid, and I have a ruler fixed so you can see the size of it. And then this is one of the skunks that's having a great time hanging out in the camera trap. So these are the results I have so far. Not a lot more to report at this point, but in the future, we're going to be able to start the experiment where we have a fully crossed design where we have half of the 60 plots will have fire ants eliminated, the other half, and so other half will not. And then also half of the plots are frequent fire and then infrequent fire. And so the idea is to determine these synergistic effects of the fire ants and the fire return interval in wildlife populations. And the ultimate goal is to provide management recommendations to the military on when to burn, how often should they burn, should they remove fire ants, how much effort should they use to spend to these things, and also grazing as well. So ideally, we will safeguard these threatened species populations or maybe even increase them in the future. So acknowledgements for lots of people that have helped in this project so far, and I'm happy to take questions. Okay. Laura, would you like to read the question? Yes. So the question is the threatened species, have they been verified and confirmed? The answer is not really, because of the photos are, especially one of them was in a photo in a camera trap. Because of the view, it's a very fixed view, and you can't normally, when you look at a snake, you can hold it and you can see what the side of its face looks like, and because it's to the top of the head, it's a lot harder. So there is still some question. Also, the only male skink that we got had been mauled by something in a funnel trap. So I'm pretty the mammals can get in and out of the funnel traps, whereas the reptiles and amphibians can't. Usually don't figure out how to do that. And so all I can figure is that a mammal got in there and killed it and literally ripped it to pieces. So it's kind of hard to tell what it is. But it is definitely a mole skink, and I believe it's a blue-tailed mole skink. But would I swear to that 100%? I would not. Well, thank you. Yeah.
Keep it going because we've got 3 talks to go.
And it says it changed your microphone, so give me a minute when you get it on there. Start its slideshow, it should be okay. Yeah. But now give me a second to get your microphone so we can. Yeah.
Oh, sure.
No, because this microphone, it said it changed the microphone. Something else. This is USB. All right, I think you're good.
Okay. Okay, thank you, Laura. Hi, everyone. I'm Joe. I'm a research fellow here at Archbold, and I direct the Predator-Prey program. Today, I'm going to be discussing a project that has its roots back in 2015, actually. It involves a lot of camera trap data. This is the first time I'm going to be showing some of these results, so bear with me as I try to move through this somewhat complicated story pretty quickly. How are we doing on advancing? Yep. There we go.
Yeah, now that.
Okay, thank you. So the project, just a quick rundown of what I'm going to talk about. I'll give a introduction to the camera trap database we have built from Buck Island Ranch. I'll talk a little bit about the machine learning tool that we are using to process and identify those data. And then we'll look at a little bit of the preliminary results of some initial analyses we've done. As I said, the project that gave us these data was initiated by our colleague, Dr. Boughton, in 2015 as a co-PI with Jesse Lewis from Arizona State University. I want to describe how these colleagues in our program have adopted the machine learning-based approach to data management, and hopefully kind of create some awareness about how this and similar tools can be useful to you if you are, and useful to many field biologists who are using camera traps and trying to navigate the wilderness of big data. So as I said, they're a popular and cost-effective tool for monitoring animal populations. They help you evaluate animal behavior and study ecological processes that influence populations. But to answer a lot of these kinds of questions, you may need a year or more of data. And depending on the size of the array that you deploy and how frequently it's timing out to take photos, you can frequently run into camera trap. You frequently build yourself a camera trap data set that numbers in the millions. And what do you do with data? How do you take those raw data, the images, and turn them into something useful for analyses? Working? Let me go back one. Okay. So this project was originally funded with a grant from the National Feral Swine Data Management Program, which is something that the USDA administers, initiated in 2014. And with the money, Raul and Jesse deployed 44 cameras at Buck Island Ranch as part of a collaborative mark recapture project designed to help deepen our collective understanding of feral pig biology in the United States. They collared and marked pigs, tracked them and looked for the before and after effects of removal, and monitored for disease. Archbold, Buck Island Ranch, is one of 4 primary sites across the country that participated in this project. 2 of the others being Tejon Ranch in California, and the Savannah River site that owned by the U.S. Department of Energy in South Carolina. So you can imagine the volume of data that camera deployment of this size is generating. So that was 2015. In the intervening years, that collaboration has produced over 20 peer-reviewed papers, and much of which derived from data collected at Buck Island Ranch. and it's been very successful. It's fueled a continuing cooperative agreement between Archbold and the USDA and their agencies, the Animal and Plant Health Inspection Service and the Agricultural Research Service. And so that cooperative agreement allows us to continue to maintain camera database even today. But that can give you, the graph on the aerial on the right kind of shows you how the cameras are deployed. They're 1 kilometer by 1 kilometer grid. There are 44 of them. They are set to time out or go to sleep after each trigger for 5-minute period. But even with that quiet period, we are talking millions of images a year. So I don't think for this crowd, I need to explain the need, further for the need to have a tool to manage this volume of data. Working with the USDA, we began, Raul and team really is who deserves credit here, worked with their quantitative ecologists and data scientists to build a tool to automatically detect, classify, and count animals in camera trap images using artificial intelligence. And the requirements that the objectives they laid out for the tool were to build separate models that would identify the taxonomic class level, taxonomic family level, and species level. So 3 different models. You also want to be able to count the number of individuals in an image. And then finally, kind of the third major objective was to be able to smoothly and accurately apply those models to out of sample images. And what I mean by out of sample, I'll explain in a bit. I've included the citation here for the paper that backs up the camera trap detector, which is the name of this model that we're using. So the advancement of the camera trap detector tool is based on an iterative training cycle with different stages capable of kind of progressing simultaneously. That means we'll test the data's performance on a subset of data, and we'll evaluate those results, and then we'll provide training data to the model to improve it. Now, it's all in an effort to constantly improve the model's performance until it begins to give us satisfying results. And what I mean by satisfying results is really kind of up to the individual ecologist. You can set the threshold at which you will accept an ID. So in the first step, you've got data curation, where we're collecting new images, running them through to the detector to see how well it performs. That's an out of sample data set. So I'll give an example of that in a little bit. And then the next step, you're going to kind of curate a training data set to feed back into the model to improve its performance. So I want to show in a minute how the model performed on an out of sample set that we took from the new corridor observatory data that we're collecting at Archbold and at the Luca Preserve through the corridor observatory project. I want to emphasize that human review is a really important part of the machine learning process, if that's not already clear. These algorithms are really powerful, but they're certainly not infallible. So it's essential to have humans in the loop to give you kind of quality assurance. Machine learning definitely makes mistakes. And so in the image on the right, we have an example of what we call a false positive. There are false positives and false negatives. So it's hard to see in this light, but I've drawn a little red box around a bunch of pine cones. And the model thinks those are a white quail for some reason. This is an example. The smaller the image, the smaller your subject is, the more training data you've got to provide. So that would be an example of a false positive, where the algorithm mistakenly identifies an object that it thinks is there when it's not actually present. Then the next example would be what we might call a false negative, where the algorithm doesn't think that anything's present, something actually is. You can't see it at all here, I'm sorry. There's a mourning dove in the foreground, just perfectly matches with the background, that hog rooting in there. And then what you want to see, though, are the true positives, positively IDs, the raccoon in the lower right and the family of bobcats in the lower left. So I'm going to take my time here so we get this right. This is confusion matrix. So on the top, you've got the predicted class, what the algorithm thinks it's seeing. On the x-axis or the right, you've got the true class. We set the acceptance level, the acceptance threshold. We set it intentionally low at 1% to say, or actually 10, to say how is it doing? How's the model doing at identifying the class of animal in this image? And so I think that says aves. We've got mammals, humans, and empty. And so not every predicted class is represented on the true class. We just bunched vehicles in with human for our data. But you can see it's actually doing fairly well. False positive rate here is on the margin, and you can see the proportion key right here. So it's actually not doing terribly at this very low acceptance threshold. When we raise the threshold to 0.8, you see it does even better with this class, identifying class in the out of sample. So now you're getting rid of you're capturing all of your empty images, and you can get rid of those immediately. That's a huge part of dealing with this much data. Now, the next slide, I want you to not pay attention so much to the identified, to the species on the margin, but look how there's no alignment here. This is with the third model, it's a species model. You're taking a bunch of data set and showing it, a big data set and showing it to a naive algorithm. So it can't figure out for the life of it what any of these species are because we haven't trained it. So that's what we'll have to do from this point forward for the observatory data. For every site, for every camera, we have to provide it at least 200 or so images so that it begins to recognize a deer and separate that out from a cow, et cetera, et cetera. So I'll go into the ranch data a little bit. This is with now, keep that in the back of your mind. So I said about false positives. This is with a verified data set. So we set the acceptance threshold for this data set at 95%. This data set dating back to 2015, we start out where we took out all of the empties and trimmed it down to just the images with an animal in it. We have 6.7 million photos. After we remove the lowest confidence guesses, that trims down to 5.3. And then, as I said, every time the camera is triggered, it takes 10 photos. We reduce that down to 1 to get to our detection. So you've got 1 detection. Now we've got something more manageable. And then finally, we weren't really happy with what the 2019 and 2020, 2021 results look like. So we decided let's focus on the first 3 years of this work. So this is the data set we're going to look at some results for going forward. So just a couple of we're just going to show a couple of simple graphs here that are should be very digestible after all the stuff we've seen today. Obviously, there's really one dominant figure at the ranch, and no matter how many years we add to this, this will remain the case. So I'm just going to buzz right through this quickly because I only have about 2 minutes left. Once you remove the cows from the picture, you can kind of see more of the community assemblage. Included in here, we have humans detected by the cameras. And I'll also show a couple of bubble maps that just kind of depict where humans are showing up on the ranch. Keep in mind, we're keeping vehicles in this data set. So people using the 770 bridge and the main road onto the South Marsh. Most of these cameras that are popping on this map are oriented to see roads. The activity periods, I don't know what these outliers are doing. I'm not sure what people are out doing on the ranch between the hours of 1 and 5. It's probably hunting, maybe some field work. But this one's interesting. This is like we've got a daytime peak of activity. Keep going with cows. The story is they're mostly active in the day, but there's a lot of variance in when we see detections. Note in this that we've got different levels of detection. So the black dots kind of show how show where white-tailed deer are distributed, and for the pigs, there's a different level of kind of occurrence on these cameras, a lot higher. But you can see where the peaks are, especially in the South Marsh and along the east side of the East Marsh. These dale periods for deer are kind of spread out, whereas for pigs, you see them mostly active in the nighttime. You can see with the two more common mesocarnivores are using really the outside edges of the ranch and the South Marsh, oriented along these main roads. I'll skip through these. But from these kinds of sort of simple data summaries, we can begin to dig a lot further into the data. For example, in this figure, each curve represents an expectation of the relationship at each individual camera. So each curve is an individual camera, and the colors represent the habitat that it's in. This curve of best fit shows the relationship between cattle and wildlife detections at each camera throughout the ranch. The takeaway is that we have ample data to start interrogating these kinds of variances at the camera level and to begin to do more complex modeling. So we're really looking forward to that. I'll buzz through these conclusions. Okay, thank you. It's quite all right.
Sorry, I forgot to unshare it.
Rosie, happy to have you here.
Oh, yeah, thanks for the second chance.
Go ahead and you should be able to share now whenever you're ready.
Okay, thank you.
And if you could self-introduce, that would be great. Thanks.
Great. Okay, thank you. Can I just check if you can see the screen?
Well, maybe I have the wrong thing. I don't see it yet.
You don't see it yet? Okay. I believe it's in the works. I see the little tiny little thing going around and around here.
Now it's
Okay, perfect. Sorry, that took some time.
okay. What I'm seeing though, there it is. Okay, let me drag it back over here now. I have to put it on the screen for the group now. 1 second. Okay, you're good.
Okay, that's great. Thank you so much. And thanks again for the second opportunity to talk to everyone, I guess, this afternoon. So my name is Roisin Stanbrook. I'm currently an assistant professor at Bethune-Cookman University, but most of the work in which I'm going to be talking to you about for the next couple of minutes form part of my postdoc that was with Josh King at the University of Central Florida. So I'm going to be talking about valuing ecosystem functions and dung beetle diversity in US subtropical working lands. And this work mostly came out of a collaboration between UCF and Boughton in Florida International University. So what is the issue that I'm going to be speaking about? It is dung removal. And I understand that it's not something that many people think a lot about, especially even when they're out on the ranch. What I've been looking at is essentially what happens underneath all of the dung pats that are dropped and how long that dung stays on the ground and what effect it has on the forage underneath the dung pats. So you can see here from the picture, this is essentially what happens when a dung pat remains on the grass for an extended period of time without being degraded. You can see that there's kind of a burnt section of grass that occurs underneath the dung pat, and that essentially takes around 2 to 3 weeks to regenerate, meaning that this forage has been lost for cattle grazing. And this is really important because having dung beetles on ranchlands, removing the dung from the dung surfaces reduces the time that it takes this forage to regrow. Importantly, having dung beetles is going to be super important, and having a higher abundance of dung beetles is super important. And we have now discovered in the last 20 years or so that dung removal is impaired by dung beetles being exposed to antihelmintics. So that's the deworming medication that is often given to cattle on ranches. So, sorry, I'm having some issues trying to move my PowerPoint on here. There we go. So what is the challenge? The challenge is essentially that most cattle in subtropical environments drop 12 10-inch dung pats per cow per day. And this is, you know, it varies obviously between temperate and subtropical locations, but it could mean that you would end up with a pasture that would be covered in dung if it wasn't for the action of dung beetles. Additionally, an issue is that cattle tend to avoid the area around the dung pat, so they won't graze the grass around all of the various cattle droppings on the pasture. And that area can expand to 6 times the area of the actual dung pat itself, meaning that you're losing all of the grazing and all of the forage around each and every dung pat. So how do we act to remedy this? Well, we wanted to discover the role of the dung beetle population in subtropical working lands and their role in degrading dung. So firstly, to do that, we had to estimate the diversity and abundance of dung beetles in both managed and natural habitats in Central Florida to gain knowledge on what species existed where, was there seasonal abundances, at what time of the year were there more dung pats lying around, were all of the dung beetle species that we have here in Florida, of which there are over 70, were they all acting at the same time? Was there deal differences? And then our second objective then, following on from that, was to estimate how much forage was smothered by dung pat fouling. So when all of these 10-inch, 12 times a day cattle pats are dropped, what is that economic cost to livestock owners? And then to do that, we created a realistic rotation schedule to include the amount of forage gained under higher abundances of dung beetles, with the aim of actually minimizing the effects of forage loss on livestock production here in Central Florida. So this project spanned really from early 2019 all the way up to the end of 2023, with kind of a shortened fill season in 2020 because of COVID. So in 2019, we began to pick research locations in Central Florida with appropriate livestock management strategies in which we could use to survey both dung fouling and dung beetles. And then we started pitfall trapping for dung beetles using 30 pitfall traps per location per month. So this continued all the way on until 2021, where we started kind of modeling our dung fouling. And we submitted our first manuscript from this survey in kind of late 2021. And then in 2022, we submitted another manuscript detailing the dung fouling that had occurred. And we also designed an app, which will be useful to ranchers to actually estimate the economic benefit of having dung beetles on their working lands. So the manuscript, unfortunately, has been in revision, I think, for almost 18 months now. And we're now on the final stage of revision. And hopefully the app and the manuscript will be released sometime in the next 6 weeks or so. So where are these experimental locations? Well, we used Durando Ranch, which is kind of slightly north from Buck Island and butts directly onto Kissimmee Prairie Preserve. And obviously we used Buck Island Ranch as well. So within each of these locations, we kind of divided Durando and Buck Island into our managed locations. And then we used Kissimmee Prairie Preserve as our natural experimental location. And then inside these locations, we set up 40 experimental mesocosms using kind of dung models, which were around 815 grams of ready mixed cattle dung, which were then deposited either within these kind of net structures that you see here or out in the open. And we placed these kind of experimental mesocosms in open cattle pastures and in locations where it was shaded. So you can see from the map on the left, all of the dark spots on the map represent tree canopies within ranches. And so we decided to put 40 of these around each of these locations just to see how much dung was degraded over a course of 6 weeks. And what we found was that if dung is left unmanipulated, so left within those mesh tents where dung beetles weren't able to access it, it would naturally decompose at kind of around 3.75 grams a day. We found then that if dung was found in managed pastures with a really high abundance of dung beetles, it would degrade at around 10.73 grams a day. And then dung that was found in managed pastures with a lower abundance of dung beetles would decay at around 7.23 grams a day. And you can see there's kind of a huge difference between the management activities and then dung beetles' responses to actually dung degradation on these lands, which I think for myself, I found super interesting. So moving on from that, now we know which dung beetles are present where, we knew how much dung they were actually degrading, how long it took. We could move on to the second objective, which was valuing the dung beetle removal. So actually putting a dollar value on it. And so this image was generated using DALL-E, and that's apparently what happens when you tell ChatGPT to give you a dung beetle and a dung ball and a dollar sign, which I don't recommend. So the history of quantifying ecosystem services has kind of mostly focused on insect pollinators and insect-mediated pest management. And we know from past papers that have looked at this, we estimate that insect pollination contributes around $20 billion annually in terms of ecosystem services and around $360 billion worldwide. Same with IPM, it's around 6 million annually. But for insect decomposers, most of the previous work had focused on looking at the impact of dung beetles on temperate systems and using dairy cattle. No study had previously been done using dung beetles in tropical lands and on beef cattle. So how did we formulate the equations used to make the ecosystem service valuation? Well, we looked at two scenarios. We looked at dung degradation without dung beetles and dung degradation with dung beetles. And we estimated what the cost of fouling would be for an entire year. We looked at the gross income per cow, and then we looked at the total heads of cattle to actually estimate how much dung was being dropped. And that gave us this value of ecosystem service benefits, basically the gross revenue in terms of additional forage area in a dollar amount. So how did we do it? Well, by looking at the dung pats and how long they took to degrade, we looked at the physical dung pats themselves. We looked at a dung pat's thickness, its density, the area that it covered, and then we estimated using NAS data how much forage was actually being smothered and the cost of that forage in terms of money lost. So to do this, Mahadev Bhatt in FIU created this wonderful attenuation matrix that looks at the kind of history of dung once it's dropped on a pasture. So you'll see here, G1, 1 represents the first dung pat that's dropped on the first day and how it continues on throughout the pasture. And then it looks at, so G2,2 looks at the history of the second dung pat dropped on the second day. And we did this for an entire year using the data of 12 dung pats that are dropped at 10-inch thickness. And this gave us a kind of potential theoretical amount of forage that had been lost and the amount of forage that could be gained from having an increased amount of forage available for cattle. And that gave us then, in turn, the total ecosystem benefits or gross income gained due to dung beetle activity under these three scenarios. So natural dung degradation with dung beetles, the ecosystem contribution of dung beetles on managed lands, and then dung beetle contribution on natural areas. And we can see that using data just from Florida, so the number of cows in Florida per year, that dung beetles overall contribute $918,000 per year to Florida ranchers, and then in natural areas it rises to $1.3 million a year. So a reminder again, the natural areas. Yeah, sorry.
Heads up, 2 minutes.
Gotcha. Okay. So representing natural areas have that higher abundance of dung beetles, and then managed land has that lower abundance of dung beetles. So which means overall that having this higher abundance of dung beetles, it means that essentially we're gaining around 86 days a year of additional forage available for cattle. So this is an example of the app that will be available once this paper is published, and which ranchers can use to input the size of their ranch, how many cattle they have on their ranch, and then how often they rotate their pastures, plus adding how their kind of visible dung beetle abundance that they have on their lands. And we think this is really needed because we don't actually know the conservation status of the dung beetles that we have here in the US. We know them from the UK, we know them from South Africa, and we know them from lots of Mediterranean countries. But for the US, we barely know which dung beetles exist where. And we think this is really important because we need to recognize the cost of the services lost as dung beetle diversity and insect abundance and biodiversity decline as a whole. And just a quick aside, I have taken this opportunity to actually ask this audience if anyone is willing to collaborate on a manuscript. So if anyone has recently knocked down any cabbage palm trees and would like to investigate the holes in which are left, I'd be really grateful to hear from you, as we're starting a collaboration with some of the natural areas around Orlando, and I'd like to extend the study out. So this is just a quick screen grab. Apparently, when cabbage palm rots in the ground, it lets off a really noxious odor. And yeah, you're going to stop me?
But we can email us.
Okay.
On holes of remaining.
Okay, perfect. All right. So I'd just like to say thank you to Buck Island Ranch and Duranda Ranch and members of the Florida State Park Service for their help with this study.
And I think the last to go, and please hang in for her because she's hung in for us, is Betsey. And yeah. Yeah.
Thanks for giving me your time, Betsey.
Thanks, Rosie. All right, everybody, thanks so much for hanging in there. Big kudos to everyone. My brain is really full right now. I'm sure yours is too. It's been a really cool day hearing about the breadth of the research going on at Archbold. So I'm the program director of agroecology at Archbold. And my research focus is on working ranches and the interface of agriculture, ecology, and conservation. And I gave this talk at the Society of Ecological Restoration in Darwin, Australia. And it was a really great conference, and I'll share some of the stuff that I learned there about restoration frameworks. I'd also like to give a shout out to my co-authors, especially Yushi Guo. She analyzed all the data in this talk, and all of my collaborators who have contributed data to this synthesis across the years. And Gregory Saunier is here in the audience with us. Okay, so in the US, wetland restoration on agricultural lands is a widespread practice, and the US Department of Ag has a voluntary program called the Agricultural Conservation Easements Program that funds wetland restoration and purchases the development rights from farmers and ranchers. The goals of the restoration projects vary, but usually focus on improving biodiversity, carbon sequestration, and water quality. And this map shows where the current agricultural wetland projects are in the US in different colors. The blue and pink symbols denote the projects, and the other colors denote the waterfowl flyways across the US. So you can see it's a really widespread practice. In many of these wetland restoration projects, some practices continue, agricultural practices such as grazing. There can be a low level of cattle grazing or prescribed fire, for example. And the restoration of these wetlands take place as embedded in different surrounding land uses, including orchards, crops, pastures, or rangelands. So in these easements, hydrology is not the only driver of wetland restoration success, and there's a need to understand how the functions and services are affected by multiple interacting management that is implemented on the landscape. And a systems approach is needed to understand how the different taxonomic groups respond and how the functions are affected. So the focus of this talk is on a particular type of wetland. These are inland, seasonal, freshwater wetlands. They're little depressions in the landscape. They're really hot spots for providing ecosystem services. They go through wetting and drying cycles, so they have active biogeochemical cycling. There's hydrological pulses, and they have high productivity, high plant diversity with zonation, and very shallow water levels. So they're sensitive to environmental changes. And the research questions of this work were to look at how does the upland surrounding agricultural land use, the cattle grazing, and the prescribed fire, which are common practices, affect 5 major ecosystem services in these seasonal wetlands. And how do these practices interact to affect the multifunctionality of seasonal wetlands? And when I went to the conference, I learned about the restorative continuum. So this kind of helped me see where we fall on that continuum of how we're implementing restoration. So we're really kind of focusing on this improving ecosystem management, repairing ecosystem function, and initiating native recovery. And so that spans some of this remediation and rehabilitation part of the restoration continuum. This research took place on Buck Island Ranch, and I'd like to just take a moment and recognize that these are the traditional lands of the Seminole and Miccosukee tribes of Florida, and we acknowledge and honor these tribes and all of the indigenous ancestors of the region. This is a full-scale commercial cattle ranch, and we implemented a long-term experiment in 2007 to look at the factorial cross of upland land use intensity, cattle grazing, and prescribed fire. The 2 levels of management can be shown here in the light tan and then the yellow. This is improved pasture or intensively managed pasture. And then the yellow is the semi-native or low intensity managed pasture. And so we've had 40 wetlands. Half of them were grazed, half of them were fenced to exclude cattle. And on top of those grazing treatments, half of them were burned and half were unburned. And to just see a picture of this, we have found in our previous research that the surrounding land use is a major driver of wetland plant composition and functions. And these management practices cascade into these embedded wetlands. This on the left shows what I'm talking about when I say intensively managed pasture. These have been planted with non-native forage grasses. They are fertilized sporadically, they're heavily ditched, they're limed, and so they receive some soil amendments. On the right, semi-native pasture, much more of a native system, does not receive soil amendments, and you can see the wetlands embedded within these pasture types. So this surrounding land use can really alter water quality, species composition, and many other wetland functions. So when you think about a wetland restoration context, if these pastures were hydrologically restored, the land use legacy of the two pasture types would remain for decades and affect the trajectory of the wetland restoration. So in this data analysis, we looked at 3 different categories of ecosystem services. Provisioning services are the products directly obtained from the ecosystems, and we measured this with forage quantity and forage quality. That relates to beef production. Regulating services are the benefits obtained from the regulation of ecosystem processes like water filtration, water quality regulation, and carbon sequestration. And then the supporting services are the indirect services that are necessary for the production of all the other services. So the way that we looked at this was habitat quality and biodiversity maintenance. So in our analysis methods, we use structural equation modeling, and this examined the effects of the management on each ecosystem service via looking at several indicators. So in the SEM, we'll consider this as a latent variable that is represented by 3 indicators. And then we can look at how the upland land use, the cattle grazing, and the prescribed fire are affecting that ecosystem service. We also calculated a multifunctionality index, and this is a way to holistically look at all of the services provided by a wetland at one time. And we just look at standardizing each of the ecosystem services on a scale of 0 to 1, and then calculating a simple average within the same ecosystem service category. And each wetland had a calculated MF index. Then we also use linear mixed effects regressions. We did this to look at the interactive effects of land use, cattle grazing, and prescribed fire on the wetland multifunctionality. And one downfall of SEM is that it can't look at interactions. Okay, so now going into several result slides, these all kind of look the same. So you'll see the latent variable here. This forage quantity service is indicated by 2 indicator values, palatable biomass and relative palatable biomass. And blue lines represent a positive correlation, and red lines indicate a negative effect. So forage quantity was, oh, I already said that. Forage quality was represented by the phosphorus content and the nitrogen phosphorus ratio. And here we can see that as phosphorus content increases, forage quality increases. As the NP ratio decreases, you get better forage quality. What this is showing is that upland intensification, so that's the improved pastures, had a positive effect on forage quality and forage quantity. Cattle grazing actually had a negative effect on quantity because they're eating and removing a lot of the biomass. The R-square value is the goodness of fit measure of these models, and these values indicate effect size. So for water quality, 3 indicators significantly define water quality: lower total P, lower available phosphorus, and lower total nitrogen. So lower nutrients, better water quality. And not surprising, we see a negative effect of upland intensification on the water quality. But this is surprising. There was a small positive effect of cattle grazing on water quality. 3 indicators were associated with soil, with carbon sequestration, plant productivity, soil total carbon, and lower organic matter decay rate. And the only factor that affected carbon sequestration was upland intensification that had a positive effect on carbon sequestration, because plant productivity is highly positively correlated with carbon sequestration. Habitat quality was represented by water pH, less water conductivity, less non-native plants, and a metric called floristic quality. And we see, not surprisingly, a negative effect of upland intensification on habitat quality, but a small positive effect of cattle grazing on wetland habitat quality. And then finally, looking at biodiversity indicators, we looked at 3 taxonomic groups: plant diversity, vertebrate diversity, and fungal diversity. And these were each represented by richness or number of taxa, the Shannon Index and Simpson Index, which take into account number and abundance of species. And we found that cattle grazing had a positive effect on plant and vertebrate diversity, while upland intensification negatively affected plant diversity. And fungal diversity was not affected by any of the management types. Now looking at multifunctionality, on the x-axis, we have fenced and grazed. The left side shows unburned, and the right side shows burned. Semi-native are the green bars, and orange are the improved intensively managed pastures. And on the y-axis, this is the multifunctionality index. So what we're finding, I was surprised, there was no difference in multifunctionality between improved intensively managed and semi-native pasture wetlands. All treatments had similar multifunctionality in the semi-native wetlands. So the only significant results that we saw, we can look first at this red bar that's saying this improved fenced and burned wetland has lower multifunctionality than the semi-native fenced and burned. And the orange shows that the improved grazed unburned versus the improved grazed burn, fire increased multifunctionality in those grazed improved wetlands. And in green, we're comparing improved fenced burned versus improved grazed burn. And grazed wetlands in that instance had higher multifunctionality. So for restoration applications, these results are relevant to restorations on agricultural land. Overall, we see land use where the wetlands are embedded is an important driver of wetland ecosystem services that will last for a very long time as we restore these wetlands. Some of our results indicate that cattle grazing is compatible with wetland restoration. These results are in the context of properly managed cattle grazing. It's an important outcome because if ranchers couldn't graze in these easements, there would probably be less enrollment in these wetland restoration programs. So we're putting a lot of effort into understanding the appropriate grazing regimes for wetland restoration. Grazing and fire favored wetland multifunctionality in improved pasture areas. My my last point here is multifunctionality a good indicator of wetland restoration success? MF can remain the same, but the wetlands can provide different services. So you really need to look at individual services, maybe
combined with the individual service analysis, it's okay. But I think if you only use it, it's not going to tell you the whole picture. And then next steps in agricultural wetland restoration. These are the 8 principles underpinning ecological restoration. And we're doing pretty good on most of these. We engage stakeholders. We draw on many types of knowledge. What we don't have really is incorporating a native reference system. And I would really like to do that to incorporate benchmarks to compare these branch wetlands. Thank you very much. That's it.